So, good day, everyone. I am Jill Vinis Bog, and I will be discussing my thesis titled Network Pharmacology and Molecular Docking Calculations Predict Hepatoprotective Mechanisms of Bioactive Compounds in Glycyrrhiza Glabra, with my thesis advisor, Professor Gerson Cabrera. So, liver cirrhosis is an end-stage chronic liver disease that is characterized by scarring fibrosis. So, it is the accumulation of fibrotic tissue in the liver. So, it results to 2 million fatalities annually. So, it's primarily causes includes hepatitis Z, hepatitis C, hepatitis B, and alcohol consumption. Its complications results to portal hypertension and hepatocellular carcinoma, and it leads to cell death and fibrosis. So current treatments include furosemide and spironolactone. So note that both of these are diuretics, so it helps in the management of ascites. So these are for hypertension and pressure in the accumulation in the abdominal cavity. So this encourages us to explore alternative medicinal methods, including natural options like medicinal herbs that possess hepatoprotective properties or benefits for treatment. So licorice is a herbaceous plant that is native in Europe and Asia. Its main compound is glycyrrhizin and is responsible for many of its therapeutic effects. So studies have shown that its efficacy in anti-inflammatory, antioxidant, antiviral, and hepatoprotective properties. So the objective of the study is to utilize network pharmacology methods such as PPI interaction, gene ontology, and KEGG pathway enrichment analysis to identify bioactive compounds that may possess hepatoprotective properties. And this also verify the ligand protein binding by using molecular docking to visualize it to know its binding affinity to the target compound. So methodology. So we were going to start by gathering the specific bioactive compounds that is available for Glycyrrhiza glabra. So we'll be using TCMSP with specific parameters that will help us gather the best target or bioactive compounds for the study. So we are going to set parameters such as 40% oral bioavailability and 0.18 drug likeness. Next, after gathering the bioactive compounds, we will be using Swiss target prediction to gather the target gene prediction of the licorice itself. So with a probability of 70%, it will generate a list of potential proteins or targets that the compound might affect liver cirrhosis related genes. Next is the GenNet or gene cards. So this is the database for target genes for the disease itself. So in our case, target gene prediction of liver cirrhosis. So we will gather specific genes that are available in liver cirrhosis in these databases. After gathering both the target genes of the plant and the disease, we will now be cross-referencing it with the um, Venn diagram. So this Venn diagram will cross-reference the specific target genes that are both available in the plant and the disease itself. Next is the protein-protein interaction. We will be using string database to map the interactions of the specific intersecting genes to one another. Next is the gene ontology and pathway enrichment analysis. So we will be seeing what these specific genes are for. So we will be checking the molecular function, the cellular component, the biological processes, and the KEGG pathways that will help us see its potential for liver cirrhosis. Next is the cytoscape. So we will be checking the hub genes or what are the top 10 genes that are mostly interacting with one another. Next is these components target pathways network. So we'll be using Cytoscape 2 to have a visualization of the connections of each components, targets, and pathways or disease pathways. Lastly, we'll be using molecular docking to verify the specific binding site and compound of the target gene. So this is to assess the binding affinity and orientation of drugs to their targets, helping to identify potential therapeutic effects. So we will now be going to the discussion. So firstly is the Venn diagram of both the target genes of the licorice and the liver cirrhosis. As you can see, there are 64 target genes of the licorice and there are 497 target genes of the liver cirrhosis. So this intersection of the diagram shows the number of bioactive compounds or target genes that target specific genes of liver cirrhosis. So this shows that the genes are the licorice compounds may directly interact with. So those genes are shown in the protein-protein interaction network in string database. So those genes corresponds. So IL-2 is involved in cellular response. HR is a receptor in that plays a role in immune response. So its activation can influence liver inflammation. So in ABCB1 is involved in drug metabolism. AKT1 is a regulator of cell survival and metabolism that can influence hepatocyte survival and fibrosis. So ESR1 is a receptor that involves modulation of fibrosis and inflammation. EGFR is a receptor that, when activated, can promote cell proliferation. F2 is involved in blood coagulation. PYGL is an enzyme involved in glycogen breakdown. SORC is a kinase that plays a role in cell signaling pathway. MMP2 is an enzyme involved in extracellular matrix remodeling, so it plays roles in tissue repair and fibrosis. MET is a receptor that plays a role in cell growth survival. ALK is a receptor TK that is involved in cell signaling. AXL is involved in cell survival and immune regulation. And AKR1A1 is an enzyme involved in drug metabolism. So we will be using Cytoscape now to rank those intersecting genes to show what are the most involved genes are there. So as you can see in the picture, so these are called hub genes. As you can see, the darker the color, the more it interacts with the other genes. So as you can see, AKT1 and MMP2 are the ones that are most involved with interactions with the other genes. So these are highly interconnected within liver cirrhosis. So by focusing on these genes, enrichment analysis can highlight specific pathways that are significantly impacted in a condition such as liver cirrhosis. So it can reveal critical pathways or diseases involved. So we can now make use of that. So this is the KEGG pathways or the gene ontology. So firstly is the biological process. So this enrichment analysis describes the biological objectives of the hub genes involved, such as metabolic process or cell signaling, oxidative stress in liver cirrhosis. So this oxidative stress can lead to hepatocyte injury, promoting inflammation and fibrosis. So basically, the hub genes involved plays a role in how cells react to oxidative damage, which can be significant in diseases like cirrhosis. Next is the cellular component. So this analysis refers to the parts of a cell or its extracellular environment where the hub genes are active. So basically, a cellular component is where the hub genes are mostly active. So in my case, or in the liver cirrhosis case, extracellular space and extracellular region are the areas that the specific hub genes are mostly interacting with. So basically, it indicates that the genes may be important in tissue structure repair, which is relevant in cirrhosis because it is extracellular space and extracellular region. So the problem in cirrhosis, mainly collagen, is hyper produced. Next is the molecular function. So this enrichment analysis describes the specific biochemical activities of the hub genes, such as enzyme activity or binding. So it suggests involvement in signaling pathways. So as you can see, nitric oxide scientist regulator activity and protein tyrosine kinase activity are the ones that are in the top. So it is a key signaling molecule in various physiological processes that include immune response. It has fibrosis modulation too. So PTKs or protein tyrosine kinase activity are involved in the activation of hepatic stellate cells. So PTKs can also regulate the activity of inflammatory cytokines. So basically, it could influence cell proliferation and fibrosis. Lastly is the KEGG pathways, where we can see what the hub genes are available, what specific diseases or pathways are the hub genes involved. So as we can see EGFR tyrosine kinase inhibitor. So it is involved in cell proliferation and survival. Proteoglycans in cancer is where this affects liver cirrhosis by altering ECM compositions, promoting fibrogenesis and regulating growth factor signaling. So now we will be going to the targets, components, pathways. So this is a visualization of the connections of each and every nodes in this image. So each represents a biological entity. So in this case, compounds, genes, and pathways. So edges shows the connection between those that indicate interaction, such as target compound. So the image show that quercetin is the targets most of the hub genes. So these hub genes are connected to specific pathways such as proteoglycans and EGFR tyrosine kinase. So this suggests that the licorice target genes involved in this resistance pathways could help overcome and prevent resistance, making future treatments more effective. So it also suggests that the licorice compounds target genes associated in proteoglycans pathway has a potential role in inhibiting tumor progression and for enhancing existing therapies. So it can be also a good way to study for cancer. That's what it's saying. Now, lastly, molecular docking. So molecular docking binding scores of the reference compound furosemide and the quercetin. So out of all the bioactive components, furosemide, which is is a positive control, quercetin has been the one who binded the same target site as furosemide. So C2. So this suggests that it has similar mechanism of action or interaction within the same target protein. So binding affinity scores show that quercetin is more tightly binded to the target. So in conclusion, enrichment analysis shows hub genes of Glycyrrhiza glabra significantly enriches key pathways associated with liver cirrhosis, such as EGFR tyrosine kinase protein receptor pathway and proteoglycans of cancer. Quercetin shows regulation of the XL gene that can potentially mitigate its role in inflammation and fibrogenesis, so just suggesting implications for future research or therapeutic development. So recommendations is to elucidate hub genes that have more interactions to the corresponding genes as MMP2 and AKT1 and other hub genes listed. And also to perform dynamic simulations to verify the stability of the protein ligand interaction. And to perform wet lab experiments that will further verify the statement. So that is all. Thank you for listening.